Hello. Continuing on with the kidney, this is where we left off. We're on the loop of Henle. First, some remarks about what we're going to talk about. The kidney can help regulate blood milliosmolarity at 300 milliosmolarity. Now, to make sense of this odd 300, what is that? It's a pretty dilute saline. It's basically the salinity of plasma. A saline solution is 0.9% NaCl. So just think of that. That saline is like the 300, okay? The salinity of plasma, pretty close to water. So you can regulate the saltiness of the water, that 300, by pulling water and salt out of the filtrate, okay? In other words, you reabsorb the water and salt. The loop of Henle, the LOH, it uses a counter current mechanism to pull lots of water and salt from the filtrate, and we'll go through that mechanism. So this dramatically varies the urine concentration. In doing so, you can maintain blood plasma milliosmolarity at 300. So to look at a picture of the loop of Henle again, well, this is the whole nephron with the vasa recta. We're talking about the juxtamedullary nephrons that can really concentrate the urine with this countercurrent mechanism. Not so much the cortical nephrons. We're talking about these ones here, the ones that are longer, that extend deep into the medulla. How do I know it's the medulla? There's my landmark. Arcuate artery and vein. So below that is renal pyramid or medulla. Above it is renal cortex. So here's a figure from your book that shows you this relative osmotic gradient where it's kind of like 300 in the cortex, right? Or the saltiness of our blood plasma, basically. But as you go into the salty medulla, it gets progressively saltier from 300 to 1200. That's the osmotic or salt gradient that we're talking about. And it's those long nephron loops of the juxtamedullary nephrons that create the gradient. They act as countercurrent multipliers. They create a salt gradient, and we usually use the term milliosmolarity. Now, molarity, you studied in chemistry, it's a concentration. Osmolarity is like biological molarity because in biology, we're concerned with the osmosis of water. So osmolarity, molarity. Well, you create this salt gradient from something like 300, which is our saline, 0.9% NaCl, to 1200 osmolarity. So the key is they create this salt gradient with the countercurrent multiplier mechanism. So we're going to talk about first is this loop of Henle, this countercurrent multiplier. What do I mean by multiplier? You multiply the saltiness of your environment of the pyramid from 300 to 1200. So maybe closer to the top of the pyramid, it's 300, matches the saltiness of blood. But as you get closer to the renal papilla, it gets closer to 1200. So you multiply on the way down like 4 times. That's the countercurrent multiplier. But then we always have to talk about the vasa recta, which is the blood supply to this region, because blood supply, well, I think by now you know why you need blood supply to every part of the body. You want to be able to supply blood to this area without washing away your salt gradient, because the saltiness of blood is about 300. You don't want to stick it through a salty environment and have it wash away salt. So there's a countercurrent exchanger mechanism we teach along with it, so you understand how this gradient is protected and not disturbed by the vasa recta. Let's first talk about the first thing, the loop of Henle, the multiplier mechanism. Looking at this slide, I like this slide. It's very simple. They just show you one side of the limb and the other limb on the way down and on the way up. So the fluid flow is countercurrent. So don't be disturbed by this fancy word, countercurrent. It just means you have fluid flow in opposite directions. Okay, this is a hairpin loop. It's a sharp turn there at the elbow of the loop. Go down, go up, and different things happen on the limb that goes down and on the limb that goes up. So, in terms of this loop of Henle, this thin descending limb, it's water permeable. It's permeable to water, but not salt. So water leaves on the thin descending limb. That's what you should know on this loop of Henle, this countercurrent multiplier thing. So in the thin descending limb, maybe I'll use the abbreviation TDL. It's water permeable. So the filtrate loses water. It's just following the rules of osmosis. It's this, as you go deeper into the medulla, it gets saltier. So water is attracted, goes to where the most stuff is. Remember the rules of osmosis, and it leaves because it's permeable to water and just follows this osmotic gradient. So, that's why the color of the filtrate gets darker, because you're losing water, and the filtrate becomes more concentrated and you're losing volume. Let's just say it becomes more salty, more concentrated. So when you get to the elbow, the bottom of the loop there, what's the concentration of the filtrate? 1200. It's going to match its environment at the elbow. 1200 milliosmolarity. That's of the filtrate. Okay, it matches the interstitium, the environment that surrounds it. Okay, then you take a U-turn and fluid flows up. Now this ascending limb is different. Something different happens. So in this ascending limb, specifically, let's talk about where it gets thicker. Call it the TAL, or thick ascending limb. TAL for short. In the thick ascending limb, you see how they draw these dark brown lines there? They're trying to show you that it's watertight. The epithelial lining is watertight. Water doesn't leak out. So water is not going to leak out like it did in the thin descending limb. But also the cells are salt extruders. They pump out sodium. Let me note that. Cells of the TAL pump out salt, NaCl. So the word pump implies they expend ATP to do it, right? So you're not losing water, but you're losing salt. So you're diluting out the filtrate. I'll say redilute. Okay, it's concentrated at the elbow, and they starting to lighten because you're pumping out the salt. And basically that's it. Lose water, lose salt, lose water, lose salt. And what does this do? You multiply the gradient from top to bottom from 300 to 1200. Now, it may be hard to kind of like see that, but what you got to do is study this positive feedback loop of what's going on by filtrate constantly flowing through and losing water on the way down, pumping out salt on the way up. This is what happens. This is the multiplier mechanism, as we call it. This is a positive feedback loop. It just keeps going on until you multiply from top to bottom, 300 to 1200. And you can start anywhere in the loop. I like to start here. They like to start there, but I'm giving the lecture. I want to start here. It doesn't matter where you start because it's a loop. You'll end up there at some point. I like to start here because this is kind of where the filtrate begins to flow through the nephron loop, through the TDL. Water leaves the descending limb on the way down. That's where I want to start. TDL, thin descending limb, right? That's the first thing I said. And what does that do? They say it increases osmolality of the filtrate in the descending limb. So I'll just paraphrase. I'll say increases saltiness of filtrate. Next point. Okay, so what does that do? So you get salty there at the elbow, and it says you have increased osmolality of filtrate entering the ascending limb. So what I'll say, to paraphrase, is you have more salt to pump out when you get to the TAL. It's kind of my next bullet point. All right, so then what? So you pump the salt out, TAL. TAL cells, pump, NaCl, our salt, out. Okay. By pumping the salt out, you're increasing the saltiness of the medulla. They say increase interstitial fluid osmolality. I'm just saying you're increasing the saltiness of the medulla, a different way of saying the same thing. So if you've increased the saltiness of the medulla because you're pumping salt out, feeds back to that. That pulls more water out. To this first point, right? It's more salty, pull more water out. Increase the saltiness of the filtrate, right? Because more water is leaving. You have more salt to pump out. The TAL cells pump out more salt, increase the saltiness, pull more water out. It just keeps going around and around and around and around. That's the loop. And that's how this nephron loop creates this gradient from 300 to 1200. And all the while, you're pulling out water and salt, which is something you want to conserve in the body. Okay, so you're pulling those things out of the filtrate. So take a picture of that. So the net effect of this positive feedback loop is, well, you just multiply the saltiness from 300 to 1200. That's it. And we've already said that, and that's all you got to understand. This positive feedback loop, it creates this gradient. Okay. So how does the vasa recta supply blood and protect the gradient? Just leave it alone. Don't wash away the salt. Let's talk about it. So the vasa recta is a countercurrent exchanger, not a multiplier. That's the key word. Don't confuse multiplier with exchanger. So the vasa recta is supplying blood without disturbing the gradient that was established by the loop. And it just makes passive exchanges with its environment. Vasa recta makes passive exchanges with the medulla without disturbing the salt gradient established by the countercurrent multiplier, the LOH, right? So these keyword passive exchanges, water and salt just enter and leave based on diffusion and osmosis regular rules of biology. So let's talk about this. What do we see here? First of all, it's countercurrent. So this blood vessel, this capillary takes a hairpin loop, just like the loop does, the loop of Henle does. This is a blood vessel. It's supplying oxygen-rich blood, red, changes from purple to blue. You have gas exchange at the vasa recta. You also have gas exchange at peritubular capillaries. I think I mentioned that already. You do not have gas exchange at glomerulus. I think I said that already too. You should know that. So what do we mean passive exchanges? Well, just rules of osmosis and diffusion. The salinity of blood plasma is about 300. Blood's coming in oxygen rich, about 300. So on the way down, you're exchanging, having your regular gas exchange, right? But notice salt is entering, water is leaving. Why is salt entering? Okay, so let me write this down. So I'll just say on the way down, blood on the way down is coming from the efferent arteriole into the downward part of the vasa recta there. Water leaves, salt enters. NaCl enters. Water leaves. Why? The blood, as it goes to a more salty environment on the way down, it's less salty than the environment it's in. So sodium wants to flow into the blood vessel, and it can because it can do it passively. Remember, capillaries are very leaky, so things can just go through the capillary wall by rules of, in this case, diffusion. So salt enters a less salty blood all the way down, right? That's why the saltiness of blood increases. Okay, and also water leaves. Why? Because as you go through a more salty environment, water gets pulled out because blood is less salty than the environment you're putting it through. So that's why salt enters, water leaves, and so basically the saltiness of the blood at the elbow of the vasa recta is 1200. Milliosmolarity. It's just matching the environment that it's in by making passive exchanges. It's equilibrating with the environment it's in. That's not so hard to understand. But now, because it's a hairpin loop, it's going to go up and out. So on the way up, talking about the blood, now you have salty blood, and it gets progressively less salty on the way up. So on the way up, water enters, salt leaves. Just the reverse by rules of diffusion and osmosis. Salt leaves the blood, water enters. So what does this do? This basically redilutes the blood there's enough salt that leaves, so the blood that leaves is almost as salty as when it enters. That's the net effect. Blood leaves almost as salty as when it entered. You didn't remove salt. That's the point. So therefore, you've protected the gradient, okay, because you didn't remove salt. Imagine if this wasn't a hairpin loop. It just flowed it flowed straight through the pyramid. It would take that salt away. It'd wash it out, okay? But it doesn't wash out because it kind of like goes back up and out, makes passive exchanges on the way down, just as it does on the way up. You're okay. You don't remove hardly any salt to disrupt the gradient. Okay, you've supplied blood and net effect, you didn't remove salt, thus protect the salt gradient. And that's really the whole point of the countercurrent exchanger. Make exchanges that leave well enough alone. Don't mess with that gradient that was created by the nephron loop. That's really the goal of the countercurrent exchanger. And let's move on. So after the loop of Henle, the next part is the distal convoluted tubule. So after loop of Henle, we come to the distal convoluted tubule, more simple cuboidal cells that line this distal tubule. And by the time we get this far into the nephron, we've gone through the renal corpuscle, PCT, loop of Henle, now we're at the DCT. There's not much left in the filtrate. Well, let me just put it this way. What's left? I'll just say a bit of water and salt. So the question becomes, do you want it? Do you want to conserve it? Do you want to let it go to the urine? Well, if you need to facilitate more sodium reabsorption, this slide, which we learned before, is the action of aldosterone. Aldosterone targets the DCT, okay? So review aldosterone. Here's a good review question. What secretes it? It does come from the adrenal cortex, zona glomerulosa. Okay, if salt levels are low or blood pressure is low, go back and review aldosterone. It's released and it targets the DCT, right? We're in the lumen of distal tubule. So you knew it targeted the kidney. Now you specifically know it targets the DCT of the nephron. You should review these physiology steps. You learned them in the first test. So basically, aldosterone is a steroid. It binds its receptor in the cytoplasm. It facilitates the production of these salt channels and pumps. Basically, facilitates sodium reabsorption. Facilitates means causes. Now, I say here, water does not follow. ADH must be present, and it will be, okay? So, water does not follow. If ADH must be present, I'm saying it will be, so that's why I wrote it here. Water does follow salt. ADH and aldosterone, they kind of always work together to accomplish the water reabsorption that's necessary. So by the time we get to the collecting duct, we are down to discussing whether or not you'll reabsorb more water to concentrate the urine or form dilute urine. That's basically what I'm going to focus on for the physiology of the collecting ducts. So the formation of dilute or concentrated urine is, well, basically, is ADH present or absent? And that'll determine it. Okay, and let's talk about those aquaporins. Again, another review slide from endocrine. So in the collecting ducts, why are they called collecting ducts? They're collecting filtrate from several nephrons. This is your last chance to modify the filtrate before it is excreted as urine. And one of the last things you can do is basically reabsorb water or not. Maybe. I'll make it a question mark. If you're well hydrated, don't need to. You can increase the urine output. If you're dehydrated, you want to reabsorb every drop that you can to conserve it in the body. You don't want to lose that fluid volume to the urine. So this is why it's the formation of concentrated or dilute urine. So you need to review the action of ADH because you still need to know that for this test. What you learned before is that ADH, or vasopressin, right? It's a protein. It's not water soluble, so it has to act through a second messenger. It's binding a membrane-bound receptor, increasing the second messenger level. And your what says exocytosis of vesicles, what you're doing is you're inserting the aquaporin, water pores, in the luminal side there. So I kind of zoomed it in there. I know it might be hard to see me pointing like that. So I zoomed in on these aquaporins. Those two little dots represent the aquaporins. And so when this is inserted, you got your water pore that can go through. So to go back to this slide, once you got a water pore in there, this is watertight right there between the cells of the collecting duct. That's where we are. It says collecting duct. Filtrate about 300 milliosmolarity. The intracellular concentration about 600. And remember, vasa recta, they say it's about 700. Remember, vasa recta is making passive exchanges, and so 700 means it's going to be a range of 300 to like 1200. So this is 700 somewhere in the middle, right? You're just going to follow rules of osmosis if you have the aquaporins inserted there, and you'll reabsorb the water, okay? With ADH, you have increased aquaporins, increased H2O reabsorption. That's what you see in this figure. And that increases the blood volume and the blood pressure there. So let's look at this slide here and just compare. The renal physiologists have done the math for us, so we can kind of put some numbers to this dilute concentrated urine. So in this slide, we have a side-by-side comparison. Here's the formation of dilute urine. You're overhydrated. Here's the formation of concentrated urine. You're dehydrated. You want to conserve water. You want to reabsorb most of it. And so the physiological range of how dilute, how concentrated you can make it is here. So let's compare overhydrated versus dehydrated. If you're overhydrated, what do we take note of? Well, no ADH, decrease aquaporins, AQP, right? So what does that mean? Increased urine output, dilute urine. That's what it says there. What says large volume of urine? Okay, it says large volume of dilute urine. And so maybe 6 liters a day, maybe each of these cups represents a liter. 6 liters a day, that's a lot of urine, and it's very dilute, maybe a 100 milliosmolarity concentration. Compare that to if you're dehydrated, well, you're going to increase ADH, increase aquaporins, AQP, so you have a small volume of concentrated urine. That's why you see more water leaving on the way down there, not so much here. It enters 100, leaves 100. Here, you can concentrate it as high as 1200 milliosmolarity. So maybe the output would be 0.5 liters per day at about a concentration of 1200 milliosmolarity. So that's the range of physiological function that the kidney can do, concentrated or dilute urine. And study urea. Urea recycling reinforces the concentration gradient to pull water out in the presence of ADH. So I'll just put no urea recycling and refer to this slide. Urea is a metabolic waste product, but the nephrons recycle urea to help reinforce the medullary gradient. Urea enters the thin ascending limb right there, okay? In the cortical collecting duct, way up here, urea stays in the nephron because this area is more dilute, but deep in the medulla, where it's highly concentrated, urea leaves, forming a pool of urea that then recycles back into the thin ascending limb, and it just keeps going on and on. It just enters, exits, enters, exits, enters, exits. You have this urea pool that helps pull water out. ADH increases urea recycling. So the next thing we do is to talk about, well, what we just talked about were the principal cells. The principal cells are for the water sodium balance. They have the aquaporins. They're the chief cell. They're the most numerous cell lining the collecting duct for the water reabsorption. So still in the collecting duct, talking about collecting duct cells, basically two types. The principal cell of the collecting duct, they have the aquaporins. Now, intercalated, stuck in between the principal cells are the intercalated cells. Inter means between. They are for the function of acid-base balance. Okay. And there's the type A and the type B. If you kind of look at what they do, they kind of flip-flop. They do the reverse. So if you kind of look at this, the type A, the type B, if you're a type A cell, here, say this is a cell, and if versus a type B cell, so on this side is filtrate, and this side is blood, okay? And on this side is filtrate, right here, and this side is blood, on this side. And so for the type A's, they got the proton pumps on the luminal side. So basically, it's going to pump out the proton, and there'll be some kind of buffer to glob onto, right, that we talked about before. So you excrete the proton, and on the other side, you reabsorb the base. You pump out the bicarb on the other side. So you reabsorb, excrete, correct? So what is that going to do when you excrete the acid, reabsorb the base? That'll help compensate for acidosis. So these cells function in acidosis, okay? The type B cells, you just rearrange the furniture, excrete the base, reabsorb the acid. Excrete base. Reabsorb the acid. So these cells function in alkalosis when pH is getting too high. And that's basically what you got to know there. So what kind of cell is this, the type A or the type B? Give you some time to think about that. This is the filtrate, that's the blood. If you're excreting the base, reabsorbing the acid, that's this one here. So that's a type B cell. All right, let's move on. Let's talk about ureters. Ureters are a muscular tube that takes the now formed urine from the kidneys, transports it down to the bladder. So their entire descent on the back body wall, they are retroperitoneal. You try to visualize them, a line on your back, using this figure. They're about 5 centimeters lateral to about L1 vertebra. So if you feel like rib 12, it's kind of close to T12. You can feel the bottom of rib 12. You reach back like that. And kind of where you reach back is about 5 centimeters from the center of L1. And I don't know if you ever like have a patient reach back. Oh man, it hurts right down here. Oh, it might be a kidney stone. Kidney stones are calcium oxalate crystals. I'm not going to test you on that, but if you eat a lot of foods high in calcium oxalate, um, rhubarb, tea, chocolate, there's some other things you can look it up. They may form, they may crystallize. And the lumen of the ureter is only 3 millimeters. So if that little pebble, those kidney stones, if they're bigger than that, it'll occlude it and it'll cause a lot of pain because there's peristaltic contraction that'll happen. Well, anyways, getting back to this, they're retroperitoneal. So imagine a line from about 5 centimeters lateral to L1 vertebra. So that's this top part here, that's the top of the ureter. All the way down, that's the posterior superior iliac spine, if you remember your anatomy, right around there. A line. So this is 1 point, 5 centimeters lateral to L1, connected to posterior superior iliac spine. So I just want to give you anatomy right around here. That's where your readers are, basically. If you want to get a imaging view of it, you can insert some contrast dye and get an image done, and you can kind of see this picture here. What we see in this muscular tube from this picture is there are some constrictions of the tube that are normal. So I'll show you those. If you look at the top, you see these three black arrows. So these black arrows, they indicate the renal papilla bulging into the minor calyces. Remember the renal papilla, that's the first place you got urine. The minor calyces collect the urine. The red numbers, 1, 2, and 3, they show you the normal constrictures of the ureter that you see. Sites of normal ureter constrictions. I see a constriction right where it connects to the renal pelvis, number 1, so at the renal pelvis ureter junction. Basically at the top. Number 2, you see a constricture where it crosses the iliac artery, the external iliac artery. You see a constricture number 3 as it traverses the bladder wall. All right. So you get all the way down to the bladder. The urinary bladder is the muscular space where you kind of house urine until you got to go. So you can hold uh, about a liter of urine in. That's about the max value. You'll start to feel urine in your bladder at around 200, 300, 400, especially 500 ml. So we're going to talk about the neural reflexes of holding it in or voiding your bladder. So do we void the bladder or do we hold it in? Voiding the bladder is just going, and holding it in is, we kind of call that your storage reflexes. So here's the anatomy of the bladder urethra. And if we cut it open, obviously we can see this is the male urinary tract. So we got a urinary bladder cut open right there. We can see prostatic, membranous, and spongy urethra, as we learned about in repro. So if we kind of look at the urinary bladder, the muscle of the bladder wall, which is smooth muscle, is called detrusor. So now we're in the pelvis here, right behind the pubic symphysis. That's where the urinary bladder is. All right. So if you cut the bladder wall open and examine the muscle inside, I'll use red for muscle to symbolize smooth muscle inside there. Basically, it's the muscle of the urinary bladder. And you have the urethra. About 8 inches in males, about 2 inches in females. And there's a couple of sphincters that help you hold the urine in. As the detrusor muscle fibers approach the orifice of the urethra, they form what's called an internal urethral sphincter. I'll just kind of draw it like this. These kind of rings going around the proximal portion of the urethra. Internal urethral sphincter. It's also like the true smooth muscle. And that's important because, there are some autonomic nerves we got to talk about. Now, there's also an external urethral sphincter. So here's the internal urethral sphincter. Here's the external urethral sphincter. It's a part of the UG diaphragm. Okay. So on my picture, maybe I'll draw it here, right? Just put some red rings going around there. Sphincters pucker. These circular muscular rings, when they pucker, like pucking your lips, okay, they tighten and they help hold the urine in. So you got an internal one and an external one. However, the external one is voluntary muscle, skeletal muscle. At the base of the bladder, you have the bladder trigone. Okay, so I'll draw two little circles here. So you have the ureters coming in, right? They're bringing in the urine, and we go behind there, and another one. So these are the ureteric orifices because you got two of them. Boom, boom. Because you got two kidneys and you got two ureters. And the third point of that bladder trigone is right there. Okay. It's the orifice of the internal urethral sphincter that makes the third point. So I'll make a little circle there. Orifice means opening of the internal urethral sphincter. And connect the dots. And you got bladder trigone. Triangle. So I want you to know that the bladder trigone is made up of these two ureteric orifices and this one orifice of the internal urethral sphincter. It's important you know that this bladder trigone, it's at the base of the bladder, basically at the bottom. So your bladder fills bottom up, okay? And another important detail to know is that these ureters, they have to pierce the bladder wall, and they go through at an oblique angle. Let me show you what I mean by that. Well, basically, if here's a wall and you got to punch through it, um, basically it doesn't punch through at a perpendicular angle. Okay, it doesn't do this. It doesn't like punch through at a straight angle there. Okay, because as the bladder fills with urine, it stretches the wall. And if it stretches the wall and it's at a perpendicular angle, it won't shut that orifice off. And you don't want urine backwashing up your ureters. So you want to enter at an oblique angle. It's more like an oblique angle like that. Okay, it's not a 90-degree angle right here, right here. It's more and an oblique angle, so that when the bladder wall stretches, it's going to pinch off that orifice. And that way the urine in there will stay in the bladder. It will not backwash. Okay. That's an important detail to understand why the urinary bladder is good for storage reflexes. Even if it's really full, it'll be full, but at least it won't backwash up to your kidneys. Now, I think there are clinical cases that where that can happen. And you can look those up. You're not responsible for them. Enter at an oblique angle, not a perpendicular angle, and keep this. Um, you keep the urine in here. Okay. And like I said, you'll start to feel bladder fullness at around 100, 200 mLs. And as you kind of fill more and more, the urge to go will become more frequent and intense, as we've all experienced at some point, I am sure, like a long car ride and it's 20 miles to the next rest area and you got to hold it in for a half an hour or whatever, that kind of thing. I remember when I was a college student, we were on a field trip to San Francisco, and we were going to the aquarium, and I had to go, and I had to hold it in for a couple of hours, and oh my gosh, I was like hunched over, and well, anyways, what happens is I shouldn't have erased my whole bladder, but just get a quick one. You got your internal sphincter and your external sphincter, right? Got your bladder trigone, the base of the bladder. As the bladder starts to fill with urine, there's these sensory signals, these afferent volleys, they call it. So at about 100, 200 ml, you quote unquote feel the urine and bladder. So you send afferent volleys to the brain, to your CNS. Say, hey, at some point you better go, okay? And they become more frequent and intense as you approach 1,000. That's your max capacity that you can hold in there, okay? As the bladder fills and fills and fills. And well, you can help holding it. You strain, right? You can receive conscious input from the brain because remember, the external sphincter can receive brain influence. So with conscious effort, you can squeeze that one just to help hold bladder, your bladder, your urethra shut, so you keep the urine in the bladder. Is what I'm trying to say for storage reflexes. So I want to study these storage reflexes and the voiding reflexes in great detail. And well, I just want you to know the nerves that make this all happen. So that's what we're going to talk about next. So this is the male anatomy, and the female anatomy is the same, except we have a shorter urethra, making females more prone to urinary tract infections than males, simply because the bacteria have a shorter distance to go to get up there. And well, here's the external sphincter, there's the internal sphincter, everything else is the same. And before we get into the reflexes, let's just talk about the nerves that would be involved. I'll go through these. So I made this outline slide to just review the nerves you got to know for the bladder reflexes. And so the afferent division, that's our sensory division, correct? So we can feel things. And in this lecture, we want to talk about the feeling of fullness of the bladder. Those are the pelvic nerves. Sensory function, you can feel, quote unquote, the bladder is full. You can decide if you want to just hold it in or you want to go, okay? But you got to feel that your bladder is full. So remember, sensory nerves, they have that pseudo-unipolar design, the short process and the longer process. The shorter process is like the central process. The peripheral process is usually longer. So they have that structure. Also remember that for the motor division, the efferent division, we're going to talk both somatic and autonomic nerves because we have both are in play. So for the efferent division, we have pudendal nerves. The pudendal nerve is part of the somatic nervous system that innervates skeletal muscle. So remember, that's going to innervate our external urethral sphincter. External urethral sphincter. So if the pudendal nerve stimulates the external urethral sphincter, sphincters tighten, that they contract, and basically that helps you hold it in. So when it contracts, we usually use a positive sign. Positive means stimulate, contract, helps you hold it in. It pinches off the urethra. Hold it in, right? You also have, innervating other things, your autonomic nervous system. You have your sympathetic and parasympathetic divisions. I have green here. For the sympathetic division, we're going to study the hypogastric nerves. Now, these efferent nerves are different. Let's remember that for the somatic division, the cell body is in the ventral horn of the spinal cord, and it's multipolar, right? It's got dendrites and whatnot, but then it has one long axon, this multipolar design. That's the design of a somatic motor neuron as opposed to the sensory neuron here. So somatic motor sensory, right? You should remember that. Take a picture of this. Continuing on with our outline for the autonomic nervous system, we have two divisions, sympathetic, parasympathetic. So for our autonomic nerves, no, hypogastric for the sympathetic division. So for sympathetic, it's like the first preganglionic one is a little bit shorter, synapses to the postganglionic one whose axon is longer. That was kind of the efferent arrangement of the sympathetic division. Now the sympathetic division, it helps you hold it in, okay? Or storage reflexes, keep the urine in the bladder until you get to the restroom, correct? And so they're going to innervate a couple of things. The internal urethral sphincter and detrusor muscle of the urinary bladder. If you're going to help hold it in, you want to inhibit, I'll put a negative sign, inhibit detrusor because that's a muscle that will squeeze the urine out of the bladder. And you want to stimulate, plus sign, the internal sphincter to shut off that tube, the urethra, which transports urine to the outside world. You want to pinch that shut. This will help you hold it in. So the nerve that helps you go, void the bladder, is the parasympathetic division. Know the pelvic splanchnic nerves. Always have trouble spelling splanchnic. Well, they'll do the reverse. Okay, these help you void your bladder, empty it. So they're going to inhibit the internal sphincter and stimulate the detrusor. So I'll just kind of keep that there. So for voiding the bladder, you want to do the reverse. You want to inhibit the internal sphincter and you want to stimulate. So make the connection. I'm using color code here. Red is for what this nerve does. and the black is for what the hypogastric nerve does. Void versus hold it in, and we'll go through it on the next slide here. So study this one. I'll outline on the whiteboard for what you got to know. Either your storage reflexes here, or Pontine Storage Center, or Pontine Micturition Center, which will help you void your bladder over here. So the brain that's involved is the pons, and what we're going to do is go through that. So we got to talk about the part of the central nervous system that's involved. So that's why they have the pons cross section. We have to talk about sympathetic efferents. So that's why we have a slice of lower thoracic or upper lumbar here. In the parasympathetic division, that's why we have part of the sacral division here. So brain, sympathetic, parasympathetic parts of the central nervous system. Okay, we've got our bladder here. We have the detrusor within there. We have internal sphincter, external sphincter, and that little square slab surrounding the urethra. So let's say you feel your bladder is full. You got to hold it in. What are those reflexes? Quote unquote, hold it in. One thing you should know for the storage reflexes, something called the somatic loop. Remember somatic loop, so that's our skeletal muscle, external urethral sphincter. There's a reflex that happens that instinctively helps you hold it in, and the loop is here. Here is our visceral afferent. Those are our pelvic nerves, right? So they go in, cross over to here. There's one that goes up there, but I'm following this one that goes directly onto the pudendal nerve, and that nerve directly innervates the external sphincter. So that's the loop. This one here, this afferent nerve, right, or cell. There's a branch that goes up, but I care about the one that goes in the spinal cord and synapses directly onto pudendal nerve. That stimulation right there. So this is the loop right there and right back out to the external. That's the loop. So that loop, the net effect of that is you're stimulating the external sphincter. That helps you hold it in. Stim, external sphincter. That helps you hold it in. And the good thing about this loop, it receives input from the brain. It's got, in other words, it's got brain influence. This is you straining to hold it in because it's the long bus ride situation. You got to hold it in until you get to the rest stop. And so here you see the Pontine Storage Center overstimulating that to just really help you hold it in. So I'll just draw another one here, another positive stimulus from brain, boom. So you have this kind of dual input, the loop, but also receiving input from the brain. Know that. And what's the other thing you got to know? Know this other one going up there, synapsing onto hypogastric. So that plus sign, you stimulate hypogastric. What does hypogastric do? Also, separate from the somatic loop, also stim hypogastric nerve, Green isn't the darkest color, but that nerve is green on the figure, so I thought I'd use it. When you stimulate the sympathetic hypogastric nerve, inhibit detrusor, stimulate the internal sphincter. That's what I mentioned before. Okay? So plus sign for the internal sphincter, internal urethral sphincter. I'm just doing internal sphincter for short. Inhibit detrusor. And that's basically what you got to know for these hold it in reflexes. Don't forget your nerves. I should probably label them. These are our pelvic nerves for the visceral afferents in blue. This is pudendal in red. That one's going to stimulate the external sphincter. Hypogastric is going to do the rest. So let's say you get to the restroom and it's time to go. Study this figure here. How to void your bladder. So what are the neural reflexes here for voiding the urinary bladder? Well, start with the afferents. The visceral afferents, they're going to stimulate the pontine micturition center. Pelvic nerves, the afferents, afferent division. They stimulate the efferents of the pontine micturition center. Pontine meaning pons, right? Now these parasympathetic fibers, what they did was they, well, just follow the parasympathetics. Once you stimulate that micturition center, follow the three results of the parasympathetic efferents. The first thing I see here, this efferent division right there, you inhibit the inhibitory, right? You inhibit hypogastric nerves. Because remember, hypogastric is what helps you hold it in. So it kept you from going. So you want to inhibit the inhibitory. So inhibit the nerve that was inhibiting the bladder from contracting. Let's follow it down here. Look at this right there. It stimulates the pelvic splanchnic nerves, and those are going to help stimulate detrusor, inhibit the internal sphincter. Stim the pelvic splanchnic nerves. Okay, yeah, those are going to stimulate plus sign, detrusor, inhibit, negative sign, internal sphincter. Number 3, inhibit pudendal, because pudendal contracted the external sphincter. So you inhibit that one. So inhibit pudendal nerve. The effect of that is to inhibit, negative sign, the external sphincter. So we've done all the work. Inhibit your sphincters, stimulate detrusor, that's going to help you void. Okay. And there's other things that happen. You contract your, when you go pee, you contract your abdominal region. That helps increase the pressure on your bladder. But this is the basic nerve function I want you to know. So we're going to move on. And, in the last lecture, here's where we'll pick it up. I'm going to stop here for now. We're going to do some sleuthing. We're going to use some blood values to determine the cause of an acidosis or alkalosis that may be happening in your patient. See you then.